We've already covered the nervous system as far as the physiology, the cells, the components. What we need to start doing now is get into the individual divisions of the nervous system. We'll start looking at the central nervous system now. We'll get into the peripheral nervous system a little bit later. Now, in the central nervous system, the brain and spinal cord, that's the only two pieces, but that's enough. They both have a very similar basic structure. What I mean by basic structure is they both have a central fluid-filled cavity. So that fluid-filled cavity is going to be filled with cerebrospinal fluid. Now, I'm hoping you're remembering back to say, okay, cerebrospinal fluid is made by... And you remember that's made by the ependymal cells and the blood vessels. The ependymal cells modify the blood into cerebrospinal fluid. And that's occurring in the ventricles of the brain. But now, around that fluid-filled space is going to be gray matter. So you can kind of see right here, it's gray matter, circling around gray matter, and this one's more like a butterfly shape, is gray matter. But all that gray matter is surrounding the fluid-filled cavity, both brain and spinal cord. But now around that is going to be white matter. Now I'm saying gray and white matter, what's the difference? Well, there is a color difference, but the reason behind it, the gray matter, most of the neurons are not covered in myelin, while the white matter, most of the neurons are covered in myelin. So that way we have the myelination in the white matter, or the lack of in the gray matter. But now the brain does have some extra features compared to the spinal cord. Sure, that's a big surprise. But you have this extra gray matter, the cerebral cortex, these different nuclei, clusters of cell bodies. Those are also found in the actual brain. You'll see here's the cerebral cortex up top, that gray matter there. So we have some extra parts and pieces on the actual brain compared to the spinal cord. So here, looking at the actual brain, so human brain, you'll notice the white matter is all down here. You can see how it looks a little whiter compared to the gray matter, which has a different tone to it there. So that's kind of the idea of white versus gray matter. White, more myelination. Gray, less myelination. All right, a few terms before we get too far. Nerves and tracts. Now, nerves and tracts, as you can see, are both bundles of axons. The difference? Location. PNS, peripheral nervous system. CNS, central nervous system. You can see the tracts are found in the central nervous system. The nerves are found in the peripheral nervous system. They're the same thing. They just change the name depending on where it is. It's still a bundle of axons. So two names, same exact structure, just based on location. Same thing with ganglia and nuclei. Ganglia and nuclei are both clusters of cell bodies, so neuron cell bodies, but it depends on location. You'll see the ganglia here is in the peripheral nervous system, while you have the nuclei in the central nervous system. Now, I know you might be thinking, hold on, nuclei, that's the nucleus of a cell. Yes, but we're using a different meaning here. So in this case, in the nervous system here, nuclei can also mean a bundle of cell bodies. So, bundle of cell bodies, looking at nuclei in the central nervous system, ganglia in the peripheral nervous system. Clustering of axons is a tract in the central nervous system, but a nerve in the peripheral nervous system. So some terminology depending on if it's central or peripheral nervous system. All right, so your brain needs to be protected. How do you do it? Don't think, what protects your brain? Well, the first thing, your scalp and your hair. You're saying, well, that doesn't do much. Think about it this way. The sun's UV radiation doesn't get through as much because you have hair and scalp protecting. Pathogens, viruses, particles, bacteria, you name it, dirt, debris, the skin can keep it out if the skin's intact. So there is some protection on the hair and the skin. Then below that's the skull. Well, hopefully you're thinking, okay, skull, we have certain bones that protect the skull, create the cranium. That's a nice, very hard, solid shell. Now that's meant more for actual impact compared to the skin. Then you have deep to that meninges. If you're getting the idea, there's a lot of protection here. Scalp, skin, hair, bone, now meninges. And there's 3 layers of these meninges. The 3 different meninges are made up of CT or connective tissue. They're meant to help protect the brain itself within the skull. And then you have this cerebrospinal fluid, that kind of watery cushion we mentioned. It helps create a little bit of buoyancy to the brain. It helps to also nourish the brain and make sure that everything's kind of helping and flowing properly. So your cerebrospinal fluid, CSF. And then we have this blood-brain barrier. The blood-brain barrier is actually a set of membranes that's going to take the blood and allow only certain things to pass through into the blood flow for the brain. It's a way of trying to make sure that you're keeping things out and only letting certain things in around the brain. So let's look at these meninges. And I'll show a picture of this in a second. But the 3 different meninges, the most exterior is the dura mater. Now the dura mater, it's kind of like a leather-like substance. It's very thick, pretty strong actually. I mean, it's not as thick and strong as leather, but if you're trying to pull at it, it's not just going to tear. You have to cut through it. It has 2 different layers of connective tissue with high amounts of fibers throughout. The fibers giving strength. You then have the middle layer, so a little bit deeper than the dura mater, is the arachnoid mater. The arachnoid mater, named from arachnoid, looks like a spider web, has lines going all throughout it. The arachnoid mater is a little bit thinner. It's not quite as strong as protective. And it has these things called the arachnoid villi. Now, arachnoid villi are important because they allow for cerebral spinal fluid reabsorption. They allow that cerebrospinal fluid to go back into the blood flow. Because you're constantly making new cerebrospinal fluid, you gotta get rid of the old. So it's a way of constantly cycling and recycling through the cerebrospinal fluid. And then the pia mater. Pia mater is the deepest of the 3. It's actually like saran wrap right on the brain. So it's gonna hug every indent, every curve, every part of the brain. If you ever did a brain dissection, you're going to see that the human brain can peel it off. Now, if you're doing different animal dissection, like a sheep brain, for example, same idea. You'd be able to try to peel it off, but it's a little tiny piece here and there. It's not just going to peel off in one big piece. So usually just cut right through the pia mater because it's so tight and clings right to every part of the brain. Now, between all this, we have these spaces. First one is the epidural space. Well, EPI, epi, the prefix epi means above or on top of. So the epidural space is above the dura mater. This is going to be found only in the spinal cord because the dura mater is right close to the actual cranium in the brain. So right to the skull there. So this epidural space is in the spinal cord only, and it's usually fat-filled in the spinal cord. I mean, it's not a huge space, but there's a little space there. Maybe you're thinking, why do I know epidural space, epidural from something? Well, maybe you're thinking about epidural injections in relation to labor. So childbirth, that's where the injection goes into that epidural space. Subdural space. Well, the prefix sub now. Sub means below or under. So in this case, the subdural space is a space under the dura mater, but above the arachnoid mater. So if you have the dura mater, arachnoid mater, this is the subdural space. Now this subdural space is a very thin layer. It's kind of filled with a very thin film of fluid. So there's not a whole lot of movement or space in here. And then we have the subarachnoid space. Again, sub, below. So subarachnoid goes from the arachnoid and the pia mater, that's the subarachnoid space in between the two. Now here though, this subarachnoid space is filled with cerebral spinal fluid. So this is where the cerebral spinal fluid is circulating around the outside of the brain. Now in the meninges, let's take a look at what they are. Let's see what they were looking at here. We have the bone up top here. The bone is technically below the skin, obviously, but it's just not shown in this model. Then we have the dura mater right below that. The dura mater, remember, is two layers. In the case, this is the brain, so it's tight to the bone. Then you have the second layer below that. You're then going to have the subdural space, the one just below that space. Now, that's only filled with a very thin film of fluid. Below or deep to the subdural space is your arachnoid mater. The arachnoid mater kind of looks like almost an orange line here. That's, again, very thin, but you can see how that arachnoid mater has this protrusion and this protrusion. Those are going to be the arachnoid villi. Now, the arachnoid villi allow for cerebral spinal fluid to leave and go back into the bloodstream. So it's helping for the reabsorption of cerebral spinal fluid. Now, just below that arachnoid layer is going to be the subarachnoid space. You can see that subarachnoid space has a little more thickness to it because that's where the cerebral spinal fluid is. It's found in the subarachnoid space. Then deep to that is going to be the pia mater. The pia mater, you can see how it's tight to the brain. It's going to follow every indent, every fold of the brain. So it's a very tight saran wrap-like covering over the brain, the pia mater. So the spaces, the coverings, the protections that we're seeing around the brain so far. But the other protection, which you couldn't see there because the blood-brain barrier is deep down underneath the brain. This is really meant to help shield the brain from harmful substances inside the bloodstream. You're saying, well, our blood shouldn't have anything harmful. Your blood is constantly fighting off pathogens, viruses, bacteria, even just dirt and debris that got in there. You have excess dead cells floating around. You don't need that getting towards your brain. So it's helping to maintain a stable and healthy environment inside the actual blood flow of your brain. And the reason it's important is it's selectively permeable. What that means is only certain things are getting through. Yes, obviously the blood itself is passing through, but it's being modified. The nutrients are being modified for a certain nutrient mix that the brain depends upon. You're also looking at modification of any drugs. So think about if you're taking ibuprofen, for example, for a headache. Well, if that's going to help your headache, it has to be able to pass this blood-brain barrier and get up into your brain. So a lot of pharmaceutical research is looking into what can pass the blood-brain barrier and therefore interact with the brain to help out like it's supposed to. So the blood-brain barrier is meant to help protect your brain and keep a lot of things out that shouldn't be in there. So the brain itself, starting with some protective measures, and we've got to move on from there.